Thank you, Oko. That's a good, it's a good addition. Okay, let's continue. That actually brings us to the first speaker. Rogier, do you want to do the introduction and then we get started from there?
Yeah, sure. I'm very happy to welcome Magdalena Harakalova, and it's a pleasure that she is here. She's a group leader in Utrecht at Utrecht University. She has a medical background as well as a scientific background, and she focuses on translational cardiovascular epigenomics, and this is just exactly as difficult as it sounds. But today she will actually explain everything concerning DNA mistakes in this specific disease. She won several prestigious prizes, and she is actually one of the most innovative and creative people I personally know. So I'm very happy that she accepted the offer to speak here today. And Magdalena, if you're ready, I'm looking forward to your talk.
Hello, everyone. Many thanks for the invitation, and thank you for the introduction, Rogier. Regarding the most inspirative people, I think I can exchange this compliment to you as well. And I will start my presentation. Let me... Sorry. So let me share my presentation. Perhaps I can just make it small. So hello, everyone. My name is Magdalena, and I indeed have a medical background. In fact, first I was focusing on neurosurgery, so I followed a surgical training. And then I was really frustrated at the surgical in the surgical room about, well, we are removing all those tumors and fixing other problems, but we are not thinking about why did it even happen in the first place, and how can we prevent it. So eventually I decided to make an experiment and basically try to see if I like more, or if I can help patients more by looking into what is actually the cause of the disease. And then I learned about how much genetics and inheritance can influence the basically diseases, and actually in many cases, including the LMNA and other cardiomyopathies, is the primary cause of the disease. So I actually started to study genetics, and I'm still very excited about genetics. I switched fields from neuroscience to cardio science, and basically started to look into how are different mutations causing the cardiomyopathy, and how can we actually help patients recognizing it, and then also how to treat it. What I'm working at the Department of Cardiology and also Regenerative Medicine Center in Utrecht at the University Medical Center and Utrecht University in the Netherlands. And the picture that you see on the front page is made by a microscope where we actually take patient tissue. This is actually a donor, a person that died because of other causes but did not have cardiomyopathy. This is how the heart cells look like. So you have a little bit of cut. This will be perhaps one cell. And what you see in the green, these are different sarcomeres. So basically the groups of proteins that actually contract and fulfill the function of the heart. And this is how they look under microscope. And the blue one...
Magdalena, sorry for interrupting, but I can't see your next slides. I'm not quite sure if Rogier and Ilko can see your next slide, but I can't see it yet.
Do you still see the first slide and my mouse?
I see the first one still, and the mouse I can see moving as well. But
correct one.
So it's correct, because I'm still on the first slide.
Okay, I'm sorry.
Thank you so much. And then what you see in the blue, this is a nucleus, and that is a very important structure of each cell, but especially in cardiomyocytes. And we will come back to that, because the LMNA gene is actually making a protein product that is very important for the nucleus. So this is just how, so that you see how cardiomyocytes, cardiac cells of every single person, yeah, mostly, hopefully look like. So I will start with DNA and inheritance. Can you please confirm that you don't see the upper panel when I'm sharing? Do you see my whole slide?
Perfect.
Now it works properly here as well.
It's only I see it. So where is actually DNA located? Because I'm going to talk about the consequences of mutations or mistakes or variants in lamin, in DNA of the lamin gene. But what is DNA in the first place? So DNA is a biochemical molecule, and you might have heard a lot about it already. But where is it present actually in our bodies? It is present almost in all cells. For example, red blood cells of humans don't have DNA. A fun fact, many animals do have DNA in red blood cells, for example, the birds. But let's say it is present in almost all cells in our body. And then where is it located in the cells? So here I have depicted a picture of a cell. Actually our tissues, our organs are composed of these little, basically living, the base blocks of our body, which are cells, and they have all kinds of organelles, which are quite the same for most of the cells. One of them is a nucleus, which we've also seen on the first slide. And where there is DNA located coming from both parents. And we have also mitochondria, which Dr. Nabben will talk about in the second talk. These are sort of like fuel factories of our bodies, and DNA is located also there. So for many people who, so that you are also realize that there are two sources of DNA in our cells. And regarding lamin, this is not located on the mitochondria. This is located in the cell nuclear structure. And when we ask ourselves what is actually the function of DNA, so DNA actually carries a code, which is embedded in these letters, ACTG, and thanks to certain combination, the cell knows whether a protein needs to be made. So how it looks like, in our body, if we really zoom in to a certain place, piece tissue, we will see cells under the microscope, and in each of these cells, we have these structures, and that I showed on the previous picture. This will be the nucleus, and these will be mitochondria. We also have other cell structures like Golgi apparatus or endoplasmic reticulum. Also, the sarcomere is a cell structure that you mentioned before, and there are little pieces of DNA in each cell, and they are called chromosomes. The chromosomes look like this, and these are literally long stretches of DNA, sort of like one long molecule that is wrapped into such piece thanks to all kinds of proteins that are wrapped around it. And eventually this sleeps. This is actually waiting in our DNA for the cells to decide, yes, I want to open that part, let's say of the LMN gene, because I need it at the moment. Let's open it. There will come other enzymes and proteins that will actually make sure that from this DNA code, we will get a sort of an RNA, which is like it's like a template, like a mirror of the DNA. And this RNA will get out of the nucleus and will make a protein. So basically, if we want to have new proteins being made, we need to have the DNA functional, and it needs to be open for us to access. So this is what's happening every single second in our bodies. Constantly new pieces of DNA or different pieces of DNA are open, and all kinds of proteins will sit on them, will make sure that RNA is made, and then eventually protein. So what will be a consequence of a mistake in DNA? As you can imagine, it will be a faulty protein. Might be also a missing protein, but like in the general definition of basically faulty, there is some sort of mistake, but that you realize that it starts on DNA in our nuclei, it will eventually be missing or faulty in the rest of the cells. And of course, that can cause these different mistakes, or faulty proteins could make us maybe have worse skin, or perhaps some DNA mutations can make us prettier or smarter, or maybe we will be shorter. But sometimes we call mutations DNA mistakes that really cause diseases, such as dilated cardiomyopathy. So what are the building blocks of the information? It is a biochemical molecule. So it is something like sugar, glucose, but it's just a really, really long one. So if you are familiar with biochemistry, or when you remember from your grammar school, from the classes, there are these chemical structures, and let's say each of the angles will be a carbon, and will have all the other attachments on it. But basically we recognize, the T and A and C and G code, which is the thymine, adenine, cytosine, and guanine, different sort of nucleotides, different chemical molecules that will combine into certain ways. And this is actually encoding the way how DNA is constructed, and also the unique combination of these four, because they have to come in repeats, or in certain repeats, they will actually tell us which gene is encoded. So the DNA of LMNA gene will consist of a lot of stretches of different combinations of these four nucleotides, and the cells have really, they know where it is, and how to make this part of DNA open and to produce the protein. So that you realize that this TACG, that's the code. And what is actually a mistake in DNA, or a variant, or a mutation? So mostly we say variant if it's something that doesn't necessarily need to lead to a disease. And mutation, if you definitely know that this variant is, in diagnostics, you might have heard from your cardiologist or geneticist that there's a likely pathogenic or pathogenic. So these ones you will likely call mutation. So coming back to the original sequence of DNA, let's say we will have the order of bases, thymine, adenine, cytosine. So there will be like a certain combination. And here you can imagine an example of a combination. But due to all kinds of mistakes, or basically due to inheritance of what actually sets of chromosomes and pieces of DNA you get, stretches of DNA you get from your parents, you can inherit a variant that will be actually different from what is normal for that protein. So for example, on this place of a thymine, we will get a cytosine. And that can have consequences for the protein in a way that it can be stopped from producing. So there is loss of protein, or the protein is incorrectly made, folded. It cannot contact other protein, or basically the structure of a cell is not the same anymore. And this is also the case for variants in lamin, of course. So how is this DNA mistake inherited? I show here like not 9, but 7 possibilities. How can you actually, as an individual, become a carrier of a variant, pathogenic variant in a lamin gene or any gene? So this is what we actually recognize in pedigrees that we construct. And I can imagine that if you actually went through DNA diagnostics, or through cardiogenetic diagnostics, you might have already thought about the pedigree of your family and where does it come from. So normally there is this consensus that squares are used for fathers, and circles are used for mothers, or for the different sexes. And if it's empty, it's healthy. If it's full, then there's a disease. We say affected, or there's disease present, and the gray might be a carrier of the variant, but there's no disease yet. And what can happen? Imagine that the mistake will be an A to G. And what I didn't mention in the previous slides is that we have, of course, 2 copies of our DNA. One is coming from the father, and what is coming from the mother. But they had also their father and mother, So eventually every single individual on that one position, so let's say, I mean, perhaps a position, basically this position, might have an adenine. So the mother can have AA, father and the mother, and the child, the offspring, might suddenly have a G on that position. And how do we find out? By looking, by sequencing, by reading the DNA using a sequencing technique. But once this is established, we know this happened de novo. That's a Latin term. That means it happened, as a novel mutation to the child. It's not coming from parents. And it's also described, it can also happen in lamin, and it is not coming from the parent. So for you to also know, it does not necessarily always need to be inherited from parents. And eventually all the mutations had to happen in the first person at some point, several hundreds or thousand years ago. So this is actually how it starts. Then we can go to, the mutation is coming from the father or from the maternal side. In the first slide, we mentioned that the nuclear DNA could come from both parents, while the mitochondrial only comes from mother, but this is the nuclear DNA. And then it can also happen that each of the mutation of the parents will have, let's say, one G. So will have only a mild disease, but the child will inherit it both, and there will be a severe disease. This is also happening in lamin. Or you can have two mutations in lamin. One will be maybe at the beginning of the protein, the other will be at the end of the protein, but the combination of both will give a severe disease in the offspring. Or it can be on the F and G. This could be on the basically the let me see if I have here a mistake. I'm sorry about that. But let's go to the G, where if it's on the if it's de novo on the X chromosome, because females have only males have only one X chromosome and females have two, it can still be that that you have a novel mutation there. But most of there are all kinds of different inheritance models that are not very frequent. But the most important for you is to know that most of the dilated cardiomyopathy in the lamin is inherited in the mode A, B, and C, and sometimes might be the other ones. So what about lamin in specifically? Because this was this far, what is DNA? How are mistakes made? Where is it located in the human body? But what is the specific situation for lamin? So as I said, the most frequently in the inheritance model, it's coming from we call it autosomal dominant, but that means that there will be one affected parent, one unaffected parent, and eventually it can give offspring or children where you can have both affected and unaffected daughters, and basically those that will have the variant have very high risk of developing dilated cardiomyopathy. So where is the lamin gene located on the chromosomes? Because we already said that the DNA information that is being wrapped into very long molecules, like if you would have stretched them, but they are wrapped so much that they are really tiny molecules in the nucleus. So this is a technique, is a result of a technique that uses different color for different parts of chromosomes. And you can nicely see here that each nucleus has two pairs of chromosomes. One is coming from each parent. And there are 23 of them. So there is 1 to 22 we call autosomes. So they are like in every single individual, and the X or Y. So here we have an example of a female that will be just it can be X and X in females and X and Y in males. But where lamin is located on chromosome 1, which is the largest chromosome. And when you look at where at the chromosome 1, it has two arms. So it is located approximately here. So this is just to illustrate that in every single cell and nucleus of your body, there is a lamin gene, which is in healthy situation producing normal protein. And it is located somewhere here on the chromosome, as I indicate. And basically, when there are mutations, if you're looking for mutations, we already know where to look, at which part of chromosome 1. So lamin AC is produced in a lot of cells and organs, basically almost in all of them. Here is an indication from Protein Atlas. It is a resource for all researchers and public to look into. That was made several years ago. And there we can look up on the RNA level or on the protein level. So as I indicated, the RNA is producing a protein of lamin. Here you see also the difference between the two sexes, and basically it's produced in every single organ. So definitely the heart, the lungs, brain, skeletal muscle, bones, intestinal organs, and the darker the red is, the highly expressed is. So you can identify the heart here, so it's very highly expressed in the heart, and the general consensus is, but it's not always the case, where the protein is being expressed, or where the RNA is expressed in protein, there you can expect a problem. And on the protein level, where it's also detected, it's almost the same organs. So in conclusion, lamin is expressed in nuclei, or like it is coming, the DNA is coming from nuclei of almost all cells. And also then it is being opened and allowed to be expressed by the machinery of that cells have to produce RNA and protein across the body. But heart is hidden a little bit here. Heart is definitely very much, lamin is very much expressed there. So where is lamin then localized when it's produced? So this seems to be a complicated picture, but what you actually see, you see actually one heart cell, or like cell. Let's say, imagine this will be a heart cell, cardiomyocyte, and this will be another one. So this is one and the other one, and this is the connection between the cells. Then inside the cells, you have the sarcomere, which is these groups of protein filaments that contract together. Here are the mitochondria, and here is the nuclear part. So this is the nucleus of the cardiomyocyte. And here is chromatin with pieces of DNA and all kinds of important proteins. And this is the membrane of the nucleus. And it has some pores where, as I said, the RNA needs to go out so that protein is produced, and also other molecules have to come in to give some information to the nucleus. For example, hormones work this way. And lamin AC is actually a part of such a structure. So lamin is actually part of the nuclear membrane, and what is shown by researchers, and will be also definitely show in the next talks, is destroying the nuclear membrane. So the consequences of lamin mutations, and now think of that lamin is expressed almost everywhere, and it's part almost of all nuclei, is that the nuclear lamina, the nuclear sort of the cover of the nucleus that keeps all the chromatin together, is disturbed. And you will hear about more of that later. And I also wanted to show you, how do we actually know that? Because often I get questions from people, and especially from patients, how do you actually know that that this is there? So what is really nice to see in the cardiomyocytes, and this is a picture from a healthy individual who died of tragic incident, and it's an electron microscopy. It's type of microscope that we use to really look at the sort of like the contrast of cell structures. And what you see here, this will be what I'm like marking with my cursor, is one cardiomyocyte. And cardiomyocytes have two nuclei. What I showed in the previous pictures, all the cells had one nucleus, but cardiomyocytes are very specialized cells that have two nuclei. And that's what you see here. So the nuclei of cardiomyocyte, of healthy cardiomyocyte, should have two nuclei. So and they should have these, each should have these two sets of chromosomes. And lamin is actually located along the black line. of each of the nucleus. And I think the researchers we are still not completely understanding what is the consequence, of both nuclei and whether one is more affected or the other one. But basically the DNA is located here. This is where the nuclear lamina is. These little gray spots, these are mitochondria, and these very regular are sarcoplasmic is sarcomere, so basically the contractive apparatus. And here you would have a vessel where blood comes and gives nutrition to the cell. So I just also wanted to show you, if you really zoom in, how does it look like? So dilated cardiomyopathy can be one of the causes of the lamin mutation, and yeah. Again, this is a cardiomyocyte, and then you have, the lamin around the nucleus. And on this picture, there are also other proteins and other structure depicted that can also cause dilated cardiomyopathy. Perhaps some of you carry even pathogenic variants in those. But I just wanted to show you, like from the cell perspective, that all of these proteins actually start with nucleus. Their DNA is there, their RNA is produced there, they will go out, the protein will be produced, and they travel to their own destination. So for example, this sodium channel protein DAP5 will travel to the membrane. The myosines or troponins or titin will travel to the sarcomeres. And lamin will come back to the nucleus and stay there. So I'd like you to think about genetic diseases of it all starts in the nucleus, and it will travel to a destination, which could be also, again, nucleus, as is the case for lamin. And my last picture is what are my last slide is about the consequences of the lamin mistakes. And it is a huge field. Lamin can carry a lot of different variants, and we still don't understand completely the fact that it's expressed in every single cell almost, and why not every single organ is affected in every single individual. There are people who only have cardiomyopathy, but also people who have all kinds of syndromes or like problems related to multiple tissues. And I think more funding is needed to still investigate what are the differences. We need you patients to commit donating blood, donating maybe biopsies, and participate in research to understand how is it possible that actually the consequences will stay in one organ for some people and will be actually generally spread across the body for other people. So this was my last slide. I hope to explain in very simple terms how do we look at the lamin variants, where are they located, and what is inheritance in general terms.
Great, thank you, Magdalena. It's greatly explained. It's a difficult topic, but you explained very nicely how the disease and the DNA structures actually work. There are a few questions. Let me see. We have a question: what governs when the disease breaks out?
That is a very. I'm sorry, my computer is now freezing, so I don't see you. But this is a very important question, because there are differences in the load of disease, not only for lamin carriers, but lamin mutation carriers, but in general. And what we know already, they are viruses can trigger, more serious disease. Has been not yet properly investigated in lamin, for example. Also, all kinds of toxins. For example, there are type of cardiomyopathies that are, or maybe some mutation carriers have higher risk to develop cardiomyopathy during pregnancy, when a lot is happening to the body, or after taking some types of chemotherapy. So in some cases, oncologists have to discuss with cardiologists whether even certain chemotherapeutic agents are even handleable, like are good for the lamin or other. I'm not sure of lamin, for example, for the titin, it has been proven. And sometimes, previously there was a lot of alcoholic, for example, cardiomyopathy. So there are all kinds of toxins and other things that can be like a second trigger, second hit to the ongoing mutation. And we understand very little about the family members who have a variant and still hasn't developed a disease. Why? What are the protective mechanisms? We are still, I think, in the middle or at the beginning of these investigations. But I genuinely think that everything that makes mitochondria, strikingly, the second topic of Dr. Nabben, that makes mitochondria vulnerable, which could be virus, which could be chemotherapy, might be the limiting factor. But these are only scientific hypotheses, and I think we need larger consortia to actually figure out.
Yeah, I completely agree. There's another question: why, or do we know why men are more affected than women?
Yeah, so that is also a very, very good question, and there are a few studies. This has not been studied properly, so we can't claim much for lamin, for example. But I showed you that through these nuclear pores, all kinds of proteins can enter, even coming from different cells, and give information. So there are these signaling molecules like hormones and androgens, the male hormones, and estrogens, progesterones, the female hormones, are also such signaling molecules that actually come to the nuclei of cells from far, and they tell something. You have to, and what they say, you have to produce these and these genes, open these and these genes and produce these and these proteins. And so what was shown that, for example, male hormones are, when they are in dish, in the lab, when they are added to cardiomyocyte, they have much more fibrotic response, so produce collagen and this whole stress response than females. That was, for example, separately established. We don't know for lamin, has to be studied still, and is being studied. And for also what's known, males have very little, they have a less flexible mitochondria. So mitochondria need to fulfill their function, switch from energy substrates, and it has been established that males or with high androgen, this works differently. So it's very, very, very important to study that, but it could very well be down to the hormones.
Yeah, great. Let's see some short answers, because now we have a lot of questions. The dilated cardiomyopathy present in the Novo variants of congenital muscular dystrophy. So is dilated cardiomyopathy present in de novo variants of congenital muscular dystrophy?
Actually, I am not sure if there is a report. I don't think that is just on top of my head, but please others correct me. I don't think that is very common. It is possible, but actually, on top of my head, I do not remember that this will be very common. Do others actually have some?
We see a lot of crossover in different, so from muscular dystrophy and lipodystrophy, we see there is also cardiac problems arising over there. But if that would be a de novo variant, that would. Yeah,
that's what I don't know exactly for de novo variants. But yeah, it definitely has been established, but not maybe Job because he's a geneticist. If he's but that is definitely a good question. Good question. It's possible for sure, but I'm not sure if such patients were diagnosed.
Let me see, we have another one. Why does the LMNA gene occur in one child and not in another child at conception? Father has LMNA gene, dilated cardiomyopathy.
Yeah, so there is a simple explanation, because the father and the mother have 2 copies of their chromosomes from their fathers and their mothers. So eventually you have 4 possibilities: the grandparent, grandmother, grandfather 1, and grandmother, grandfather 2, chromosome 1, and the child only gets 2. So if the first grandmother's chromosome is the faulty one, then it doesn't have to go, doesn't have to be the two that go to the child. So so this is how, just by random, depending on which sort of cells were used for conception, there will be the mutation or not. And I don't think anyone can influence that. This is just statistics, how the combination works. It's the same chance as having a child that has female or male sex. So it is definitely by chance, and it's not known if we can influence that, you know, biologically, eat something or do something. That doesn't work this way.
And there is a nice discussion ongoing about the evidence, or is there new medication available for LMNA patients? So is there already something on the market for the patients?
Yes, very good question, and the later talks will hint and show more about it. So I also don't want to spoil too much, but yeah, it stays difficult. But one thing stays, that the primary cause of the disease is this DNA mistake. And if we focus on correcting that mistake, it's still very difficult, but that is the future. And another, so you can focus on correcting the very same mistake. And then different organelles of cardiomyocytes, such as mitochondria or sarcomeres or the nuclear membrane, are destroyed. So if you try to correct or help them to function, the disease might come later and be less severe. So basically, we need to understand what is happening in the cell and what are the consequences, and actually target those. But there were some clinical trial recently. I'm not sure if they will be discussed, but there is definitely some progress. Yeah, very promising maybe info coming from the genetic engineering field. However, it's still not there.
It's a hugely developing field, and on gene therapies, but also this question was about the medication and the medication. So as far as I know, but perhaps Professor Duvedans can say something on that. But as far as I know, there is no medication available yet, besides the clinical trials that are ongoing right now.
I have to agree with you, Rogier. That's perfectly summarized.
And I see an interesting on exercise. Maybe this could be the last one. Time-wise, there was this, you know, the conflicting information on exercise and what to do to cell or something else caused that. I know there is conflicting information on exercise, and this is very, very good point. And we still don't understand completely how does exercise influence in between different genes. There is something published on arrhythmogenic right ventricular cardiomyopathy that exercise is actually a trigger. Our colleagues tried in other cardiomyopathies. These results are still not published. They are, you know, evaluating the questionnaires, but most of the questionnaires are asking you what kind of exercise have you done when you were a child, a teenager, or like in general. Ideally, we need to fund and do more studies that actually test more functionally, so that you sport while having all the ECG electrodes on your chest and maybe measure some blood biomarkers before and after. But then we will know, you know, better. But it is definitely conflicting, and I don't think anyone can make an advice, any association, do sport or do not sport. But my general suggestion will be, try to imagine that you need to have a life of a pregnant person. With eating, with movement, with sleeping, with stress and balance. Try to adopt such a lifestyle when you have a sort of a risk variant. And I don't think that could be anything bad with that.
Yeah, I completely agree. I think other questions can still be put in the Q&A, so we will answer them. Live. Stefan, you want to?
No, it's a lot of questions, which is great, of course. That's what it's meant for, an LMNA patient meeting. So if there's more questions, please put them in the Q&A. Yeah, so that's only a good thing. You're busy, Rogier, with answering the questions on the background as well, because I see a lot of questions coming in. I'm sorry I'm not able to help you there.
We'll be okay. I think we go to the next speaker.
Yeah, so are we doing a break first, or are we going straight to the next speaker? Because that's Yeah, and I think Ilko wanted to do a small introduction right after the break, or maybe you want to do it before the break. I'm not quite sure. What do you prefer, Ilko?
Well, we do have a small coffee break in the program now, so I suggest we keep us to the program, and
Sounds like a good
see
plan.
everybody back in 15 minutes. Well,
Yeah,
let's
now
make
we'll
it
be
10
back in
because
about
we're 5 minutes behind schedule. So
10 minutes.
yes, we will continue at 5 o'clock sharp, and that's
Perfect.
our time. So
That's great.
okay, thank you. Okay, welcome back, everybody.
Yeah, 10 minutes sharp is 10 minutes sharp, Ilko.
Exactly.
On the clock. That's great.
Maybe the panelists can turn on their videos again. That's a nice way to have this interaction. Yes, indeed, Stefan, let's continue.
Yeah, you wanted to do a small introduction from your side as well. We actually had it scheduled for the end of the LMNA patient meeting, but you want to do it in a small introduction about the Netherlands Heart Institute?
Yes,
yeah,
but,
that's great.
For sure. But let's first thank Magdalena for guiding us through the difficult and complicated interaction of genetics and proteins. I think you did a great job, Magdalena. We've had a lot of questions. You explained us how this can lead to LMNA disease, and well, hopefully later this afternoon we'll also see that it can help us find a cure. But thanks so much.
Thank you.
Yes, today we host this meeting, the Netherlands Heart Institute. For those who don't know, it's a cooperation of the Dutch academic departments of cardiology. And the goal of the institute is to stimulate cooperation in research groups. And more we do this together with patient communities. As such, we had a great first meeting in April. Stefan, you already mentioned it. We did that together with LMNA Cardiac. And we're happy to participate in this meeting as well. And maybe another point other thing I'd like to point out. One of our initiatives lately is that we have started a heart tissue bank, something I would like to mention here, because I think it can be of great importance for LMNA research as well. And we'll share information about that after the meeting. So just to make you aware of this. Yes, well, that's more about it. We like to keep it short and to the point today. So I'd like to give the word back to Rogier. Thank you very much.
Great, thank you, Ilko. So ready for talk 2. So Miranda Nabben is here. Dr. Nabben is assistant professor, Department of Genetics and Cell Biology. And she has a bachelor and a master and a PhD, and she did several postdocs within the Netherlands and outside of the Netherlands. And her ambition is to treat cardiac diseases via metabolic interventions. And what that will be, she's going to explain in her talk. And it's very nice to have her here, a colleague from Maastricht. But also when I just look back in time, then we know each other already for like 25 years. So that's a very depressing fact that we're getting old. So that's, but I'm very happy that she's here, and we also collaborate on LMNA research, and I'm looking forward to your talk, Miranda.
Yes, thank you, Rogier, and yeah, also thank you to the other organizers for inviting me to present here. So while I'm starting to share my screen, I would like to mention that indeed I'm not a medical doctor. I have a PhD, so I'm a research scientist. So whenever I say something that you think, well, that sounds very strange, just ask or comment on it, please, because I'm also not used to give a presentation to patients as well. So I try to make it clear, and hopefully it is clear, but in case it's not also there, just ask questions. Don't hesitate to interrupt or if I go too fast. So I think actually the end of the talk of Magdalena was a really nice beginning of my talk, because I have a bit of exercise included in my talk. And I'm going to talk about metabolism in LMNA, but I will start with explaining a bit more what metabolism is and how we should see it. And then I will end with some findings that we have at this stage, which are not published yet. And also I will make a small link to what is known in literature and how we think metabolism is very important and playing a role in laminopathies as well. So starting with explaining it, I usually like to see the heart of a person and then compare it with the engine of a car. And there are a couple of things, of course, very important for the engine of a car. And one of them, I think, is this, and I hope it's going to work. Are you able to see the YouTube video now, which is starting? Do you see it now? I will do a new share. I'm sorry, I will do it quickly. But I will, yeah, it's actually, it is Max Verstappen, not last weekend, but the weekend before in Singapore, where he was riding the qualification round number 3. And I don't know if you remember what happened there. I'll do it very shortly.
Losing a bit of time. Sonoda goes ahead of Gasly. Max Verstappen been on the front row here before. He's not going to be on the front row tonight. He aborts the lap, and it gives Charles Leclerc a pole position. And Verstappen on the weekend, where he could seal the championship, will be starting from 8th. Box, Max. Box, 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 box. Why? What the? Why?
Okay, I will stop so far with all the swearing that you will now hear. But does anyone remember what was going on there?
No more fuel.
Exactly. There was no more fuel. And that was actually also the important thing, because that is basically what is related to the metabolism. Your engine needs a lot of fuel, and your heart will also need a lot of fuel. And the oxidation of this fuel, that is basically the metabolism. And then, so the parts that I'm going to talk about in relation to metabolism is I'm going to talk a bit about the type and the amount of fuel that your heart will use. I'm going to talk about how the fuel gets into your heart and whether it gets burned properly. And I will start with the first one. And so again, when thinking about the heart as a sort of car, you can see it as a sort of hybrid. So the heart can use a lot of different fuels, actually. Depending on what it will get, it will start to burn it normally. And so the main types of fuel for the heart are actually your fat and the sugar. So you can get them from the diet, but you can also get them from what is stored inside your body. To lesser extent, it will be lactate and ketone bodies and amino acids. But the fat and the sugar are the main substrates. And it will burn it actually in about 50% fat, 50% sugar. Usually there's quite a nice balance there. And so basically, when you look at the healthy heart, and you're kind of in the middle. However, what we see is that the healthy heart is quite flexible. So depending on the condition that it's in, it will switch. So for example, when you think about a marathon, and I wrote down low intensity exercise, well, low intensity, that's, well, it's not really a low intensity, but at least it's a more endurance exercise. So it takes a long time. And that is when your body starts to use a lot of fat. So it takes a long time to burn the fat, but it will give a lot of energy, more than sugar. On the other hand, when you start to do a sprint and you need your energy very fast, so at a very high intensity, that's usually when you start to burn a lot of sugar. But what we see is that when you think about the fuel preference, we see that when it's chronically on one of the substrates for a very long time, so for example, in diabetes, where the heart has difficulty in taking up the sugar into the cell, and therefore it will burn a lot of fat in that case. When that's for a long time, we see that that's associated with a decrease in cardiac function. On the other hand, we see that, for example, in animals that have a high blood pressure without any diabetes or any other comorbidities, then we see that the heart starts to burn a lot of sugar, and that's also associated with a decrease in cardiac function. So it seems that when you go to either side of the balance for a very long time, it doesn't seem to be a good thing. So ideally, what we do in our group is we try to rebalance this substrate use. So we try to make it even, basically. So that is the first part about the type, and later on also the amount of fuel, because that will be important. Another thing what I would like to explain, and therefore we go a bit more into detail, is how this fuel gets into the heart. So, and therefore I would like to zoom into this heart cell, basically, in order to explain it. So what you see with a heart cell, and this is a very schematic overview where in the light yellow pinkish color shows the heart, and then the more reddish color shows the blood. You see the fat and the sugar, they are in the blood, but they will enter the cell via so-called fat and sugar transporters. And once they are inside the cell, they can be burned, and that will actually result into energy, or they can be stored. And but you can imagine that these fat transporters and the sugar transporters that are there at the cell surface, that they are very important, because the more transporters they are there, the more of the substrate of the fat and the sugar will be taken up. The less transporters are there, the less will be taken up. And what is also important there is that these transporters are not always there present at the cell surface, but they can actually move from inside some sort of vesicles called the endosomes, which are very acidic. And from there, they can move towards the cell surface and back. And that's for both the type of transporters. And this is usually regulated by hormones, for example, insulin. So that is what you see in diabetes. Insulin is very well known to increase the transport of the glucose transporters towards the cell surface. But also contraction can stimulate the translocation of these transporters to the cell surface. And then a third thing is the question whether the fuel gets burned properly, because that's also, of course, very important. And yeah, again, this picture, and I already showed you that the fuels can get burned. And this is actually happening in the mitochondria. And I think Magdalena already touched upon it, that they are very important there. And so this is a kind of a drawing. You can kind of associate it with the carburetor of the car. So it will burn the fuel, and this is actually what it looks like in real life or under the microscope. And also there, I think Magdalena already showed it a bit in a smaller view. This is a bit more zoomed in. So you have these mitochondria and you have this so-called cristae. And these are very important parts of the mitochondria, because that is where the oxidation actually of the fuels or the burning of the fuel take place. So when you zoom a bit further into the cristae, then it looks a bit like this. And you don't have to remember the details or anything. That's not important at all. But what I want to show you with it is that it's kind of complex, but it also consists of several complexes. So the oxidation of the fuels will result in also called NADH and FADH2, and they will donate protons or electrons, doesn't matter. The electrons will go through the complexes, and in the end, at the fifth complex, also called ATP synthase, that is where ATP or energy will be formed. So ATP is another word for the energy. And this process will use oxygen, and I will come back to that later when I show you some of our results as well. So the mitochondria are very important. And therefore, luckily, we don't have just one mitochondria in the cell. We actually have a lot of mitochondria in the cell. So usually in a heart cell, it's about 5,000 to 8,000 mitochondria. And this is a picture of the electron microscopy, where you see all these kind of black round things. These are the mitochondria. This is a picture of a cardiac biopsy. So we were very happy that we had this. So this is from a patient that has a non-genetic dilated cardiomyopathy, which we in this case used a bit as control. Next to it, I show you a picture of a cardiac biopsy of a mouse that is, basically completely healthy mouse tissue, where you also see the mitochondria nicely aligned. So there's a lot of mitochondria in the cell. So when looking at the total picture, then we basically see that the type and the amount of fuel is very important. So what is offered to the heart in this case? How does it get into the heart? And does the fuel get burned properly? And this is also what we investigated in our case. This is from a patient with a founder mutation, dilated cardiomyopathy. And this patient came to the clinic, and Job Verdonschot who's actually sitting with me in the same office now at this moment, but he's muted. But yeah, what he saw, like they saw these patients coming in, the patient had a decreased cardiac function, had an enlarged heart, and the patient had arrhythmias. And yeah, then I won't go into the detail on the whole story, but this appeared to be a founder mutation, so a new mutation in LMNA. And what we did was that this patient was so kind to donate blood to us. So we isolated the blood cells, and then we reprogrammed them into stem cells. And those stem cells were differentiated into heart cells. And that is what Leanne Wong, a postdoc in our group, did. And so the data that I will show you is mostly the work of what Leanne coordinated there. And we had actually 2 clones of this patient that we used. So that is what I will show you in the data. That's why you will see 2 bars for the patient all the time. So clones means that we had 2 different types of cells, stem cells, that we then made heart cells from. And so this is the first data actually. And it might look a bit complicated. That's all right. The main message, what I want to tell you here is that what we saw was that in this patient, we saw that there were less mitochondria present. So again, it's only 1 patient, 2 clones. So, but yeah, this is what we saw basically. So, and on the top, you see the healthy control, and I draw the line there. And that's kind of the amount of mitochondria on the line. And you see that it shifted towards the left. So we see that there's much less mitochondria present in these patient cells, the 2 purple ones compared to the red one. And also when we looked at, well, first in figure D and E, what you can see is that in the black bar is the control. So, well, basically the healthy person, or assumed to be healthy, of course, we never know exactly, but at least the patient that did not have laminopathy. And what we saw was that compared to the cells of that patient, we saw that there was much less expression of this mitochondrial protein in the 2 clones of our patient. And also when we looked at PGC1 alpha, which is a gene that is involved in the production of mitochondria in the body, we saw that this was lower in the patient cells. So there was less of this gene present actually, which would fit the whole picture that eventually there's less mitochondria here. So then we also looked at the mitochondrial function. Also there, it might look complicated, but that's all right. The main message here is that I told you already that in order to produce energy, you need oxygen. So often what we suggest is that the less oxygen that is being used, the less energy is produced. Of course, we cannot be completely sure because we did not measure the amount of energy. But what we saw that was that when we fed those cells actually of these patients and of the control, we saw that the patient cells were using less oxygen and probably producing less energy there as well. And also we saw that the patients, so the CL1 and the CL2, the patient cells showed less of these complexes that I showed you before, this complex 1 till 5. So there was less of this complexes present, which, and also if you look at the so-called oxygen consumption rate, so how much oxygen is being used, that was all less in the patient, which could all fit with that there were just less mitochondria present, of course, in this patient, and therefore it could burn less fuel. Then we also looked at the fuel itself. And when we looked at the fuel uptake, we see some variation there between the 2 patient clones. But for both patient clones, we saw that the fat uptake into the cell was actually less in the patient. When we looked at the fat transporter, we saw that it was not significantly less. But this is the total amount of fat transporter. So that means it includes the fat transporter that is present both at the cell surface, but also inside the cell at the endosomes. So it could be that the amount of total fat transporters is the same, but that there's just less present at the cell surface. That we don't know, but what we do see is that there's less fat being taken up. On the other hand, when we look at the sugar uptake, we see that there's much more sugar being taken up in the cells of this patient. And also when we look under the microscope and we look at these black dots, we saw that in the patient cells, compared to the control, so-called wild type, we saw that there were much more of these black dots present. And these black dots is actually glycogen, which is the storage product of glucose. So to summarize what we found, and again, I want to stress that this is only one patient and also only 2 clones of a patient cell. So we have to be careful with interpretation. But what we saw in these cells, at least, was that there was less fat being taken up into the cell. There was more glucose being taken up into the cell. There was, but there was less glucose being burned, but also probably because there were less mitochondria present. And we saw more glucose being stored. So probably the glucose could not go to the mitochondria for oxidation because there were less mitochondria, and therefore the glucose started to store. And then coming towards the final slides, and this is a very busy slide, I know that, but I looked in literature and then I tried to briefly summarize. But what we see is that in general, there are associations with lemminopathy patients where some patients do have lipodystrophy, it's called, which means that there's a sort of disturbed fat distribution among the body. So in some places there's more fat being stored, in other places there's less fat being stored. There are also association seen with diabetes, with some patients having a higher risk of becoming diabetic or insulin resistant. When we look at mice studies, we do see mice that show impaired mitochondrial biogenesis. So that's impairment in the production of mitochondria. Actually, what we also see in our patient cells that we investigated. And there's mitochondrial dysfunction being seen, which is the same which is seen in skin cells of patients with a lemminopathy, basically. So impaired mitochondrial respiration, and also which leads then to oxidative stress. So damage to the mitochondria and damage to other parts of the tissue. Also in the fat, less mitochondria. So basically in different types of disease, we see different phenotypes, metabolic phenotypes. But there's still a lot not known there, but there's, I'm very convinced that there's definitely associations with lemminopathy and associations in relation to alterations in metabolism there. So the hypothesis that we have is that at this stage, we think that's, like I just said, different LMNA mutations. And then also you should not just look at the type of mutation, but also at comorbidities. For example, in a patient, if the patient is diabetic or is having a high blood pressure. I do think that leads to different cardiac, at least, metabolic alterations. I also think whole body metabolic alterations. So that is something I'm pretty convinced of. We have to see, of course, what that means. A thing that is a bit less clear, but that's we hypothesize that is also going on, and that Magdalena also stressed already a bit, is that what I think is that if you have a metabolic alteration already, and you put then an extra metabolic stressor on top of it, for example, extreme intensity exercise or chemotherapy, I think that might trigger actually the development towards heart failure. And with exercise, I'm talking about extreme exercise. So not just moving or being active, but really extreme intensity. And the third thing what we hypothesize is that, at least what we hope, is that targeting these metabolic alterations may have good potential for treatment. But like I said, there's a lot of research that still needs to be done there. So what we do with our group is we try to get a further understanding of these metabolic aspects of cardiac disease in general, but lemminopathy is definitely one of our points of attention there. Then we try to identify novel targets for treatments. And yeah, hopefully what we aim to do, what our overall aim is to well, provide insight and eventually hope to get to new potential therapeutic strategies for treatment. And with that, I hope that I would like to finish. I hope that in the end, well, we will all look a bit more like racing cars, or have an engine that is so powerful like the one from Max Verstappen. And yeah, so basically that's our aim. I would like to thank you for your attention. I'm happy to answer any questions. And I actually do have one question to the patients perhaps in the audience. So I'm curious to hear if, for example, if you did experience, or after hearing my story, if you can relate to, for example, noticing more symptoms or having more problems after metabolic stress. So that is actually, and then again, yeah, maybe extreme exercise, or maybe it was cancer treatments, which can put metabolic stress on there. So that is actually my question to you. So yeah, thank you very much for your attention, and I would be happy also to answer any of your questions.
Great, Miranda, thank you very much. This clear explanation, and of course, it's also very nice to see some Max Verstappen analogies. So, of course, as Dutch, we are very proud to have the new world champion, or the remaining world champion. There are some questions, actually. I see that Job and Magdalena already are answering some questions. Let's see. Miranda, does COVID or certain shots a part of metabolic stressors to speed up heart failure, like intense exercise and chemotherapy appear to? So do doctors know if there is so other stressors to speed up heart failure?
Yeah, so one of the things I have to stress again is that it's a hypothesis that intense exercise and chemotherapy do so. So we are not completely, or we're not, at this stage, we're not sure. That is what we think might happen. Yeah, it's a difficult question. I'm not a medical doctor, so I'm very careful in saying that COVID or certain shots might do anything there. I do think that COVID does put metabolic stress on the body, or could put metabolic stress on the body, because it asks a lot from your body. I also think that diabetic patients were at greater risk of developing problems, at least in relation to COVID. But I think, yeah, we definitely need more research to put any conclusion there. So I'm very careful in, yeah, giving a clear answer. I'm sorry for that.
No problem. I think Magdalena answered another question as well, but I would like to have your vision on that. So then so our anonymous attendee has two expert visions on this. So is there any evidence for specific diets in cardiac and neuromuscular LMNA problems?
No, not
evidence for diets.
Not that I'm aware of. Again, same question there, and I didn't put the diet in there because I always think it's a bit tricky, and also because I think it might vary per patient what your metabolic profile is. With that I mean whether your balance is shifted towards fat or towards glucose. Like I said, it can be influenced by a lot, like if you have diabetes or high blood pressure, if your gender also influences it. But I can imagine from a metabolic point of view, if you eat a high-fat diet for a long term or a high-carbohydrate diet for a long term, it will lead to cardiac problems in general for patients. So I can imagine that also for laminopathy patients, it's can lead to problems there. But yeah, I'm not aware of any studies that really investigated this in a large scale. So.
I think there is also an answer to your question you asked to the patients. So when in physical stress, I tend to feel lethargic and crave carbohydrates instead of a more protein-based diet I normally take. So, and which is advised with the neuromuscular disorder. So that's an answer for that. So there is a different, a different feeling, a different craving for those types of diets. So that's indeed an interesting part.
Maybe if you're looking for more answers on that question, Miranda, maybe we can do a survey within our network at some point. I think it's a very, very interesting question. And I myself can tell the difference as well. So I think I can relate to that question as well. But maybe it's good to do it under a big yeah,
I would
group
be
of people.
very happy with that. If that's possible, that
Yeah.
would be great.
Yeah, I think that will be very interesting, and I think this is definitely an interesting topic for Rogier as well. We spoke about it before, so maybe this is something that we can set up and see if we can get some proper answers on this.
But you said you had some recognition over there. So what is your personal opinion? If you would ask, if Miranda would ask you, what is your answer?
I can definitely tell some difference as well, but maybe it's good to set up a proper survey that you can actually how you can actually tell. But I can definitely tell the difference if I. Yeah, I can relate to this question, and I can definitely answer on this question as well. That I can tell the difference. But I can definitely tell the difference. Let's put it that way. And I have some examples as well.
And maybe one such survey will be. Oh, just a second.
In the meantime, I also had a question, and the question is also a question which is put in the chat. So that's quite similar. Could a diet low in carbohydrates have a positive influence on the heart of someone with an LMNA cardiomyopathy? So I was thinking a lot of people are on a diet, a certain diet, or want to cut down on the carbohydrates. Would that have an effect on the story you just told us?
Yeah, so therefore I think it's also important that you're almost, we talk about laminopathies, but I think it's like almost person dependent what your metabolic profile will be. So indeed, if you as a person if your heart in this case is using a lot of, or taking up a lot of glucose, in that case, it might, a lot of sugar, I called it in the presentation, it might be good to cut down on the carbs and cut down on the sugar. But it really depends. I think it's really person dependent. And for that, it would be good to have more information first on what your metabolic profile is. Overall, I think it's good to have a bit of a balanced mix of diet.
You had an extra remark?
I wanted to also mention that in general we should be investigating perhaps things that are lowering blood sugar levels, including prolonged periods of sleep deprivation, which, for example, young parents experience with mental health problems when you have experienced stress at certain periods of your life, including viruses are depriving, glucose, or alcohol, for example, messes us a lot with glucose investigation. And we had a lot of discussion about the alcohol, especially, but they are like, we should think about things that can basically mess up with this, especially maybe glucose metabolism, and which ones are depleting them. But could be indeed, it could be also the fat metabolism. But general sort of a general solution might be not too little and not too much of either of the fuel would be the best. So the balance.
Yeah, I agree with that. I think a balance is probably the best, especially until you know what your metabolic profile would be, of course. And I think that's a stage where we are not at this stage, of course, in research. So, but
it would be nice to include the sleep deprivation and stress, mental stress, and but basically sleep is also one of the things that are is very underestimated, how important it is also for cardiac health. So just general advice can't hurt. Just sleep well if you can.
All for free, this medical advice. So that's great. I have another question, Miranda. I had a laminopathy, but I had a heart transplant, so I don't know if I have it anymore. Would the LMNA still affect me?
Yeah, that's a good question. And then I think I would have to look at a job probably with the medical doctor in the audience. Job, after a heart transplant? I have to mute because Job is in the same room as I am, but I will give the floor to him.
Yes, I think this works. Yeah, so after a heart transplantation, what happens indeed is that you have a new heart, and that specific heart you will receive does not have the LMNA mutation. So indeed, that you're a carrier of a LMNA mutation will not affect your new heart. But keep in mind that it's all the cells in your body have, of course, genetic material, so that you have a new heart is that you don't have the mutation there, but other cells in your body can still have the mutation. Why could this be relevant is, for example, if someone had a heart transplantation and still has a child wish, so still wants to get children, then of course you can still give the genetic mutation to future children. So it's not that if you have a heart transplantation that your whole body is cured from the mutation.
I have another question. I wonder if symptoms onset earlier for young adult CrossFitters.
Sorry, I didn't, was the question to me, I guess, right? Job and I were just switching the sound, so I missed the question. I'm sorry.
No problem. I will repeat the question. I wonder if symptoms onset earlier for young adult CrossFitters. So I think that relates to the stressor, the mechanical stress problem, which was earlier mentioned. So if young adult CrossFitters, would they experience the problem earlier, the onset of disease?
If our hypothesis, so again, hypothesis is not proven, if that's correct, I think it might be the case. So if you have a LMNA,
that also relates to my question, and also which was mentioned earlier, and also relates a bit to my research. So that's so extreme research, so heavy interval research, high intensity might be worsening. But I think it's so young adult CrossFitters, if you just move and just light exercise, as Magdalena called, like a pregnancy, that would be so that's still healthy, but try to avoid like high intensity training.
Yeah, that's the hypothesis indeed that we have.
Yeah. And I see that there is a very difficult question, so I'm happy that Job Verdonschot is answering the question already. Can the medicine Forxiga slow down LMNA DCM? So you want to already explain that for the whole audience, Job? Or are you just typing?
Yeah, I was typing, but maybe I can also answer live. So the Forxiga is DAPA. It's an SGLT2 inhibitor. It's one of the newest medicine for heart failure in general. So we do know that it is indicated and also included in the most recent guidelines for heart failure that it's very effective in heart failure. But it is quite a new medicine, so we only prescribe it maybe for 1-2 years. So we don't have any information yet for specifically LMNA DCM, but it will be very important in the upcoming years to collect this data and to have sufficient information of this medication use in LMNA DCM patients. Because maybe this will be, because it also influences metabolism, maybe it will be very helpful for specifically LMNA.
Great, thank you, Job. Thank you, Miranda, as well.
Yeah, can I just briefly add up on it? So what the SGLT2 inhibitors do is they lower the glucose levels in the blood. So that's basically how they work. So indeed, you can imagine if there is a lot of glucose that can, well, in that case, or there is a lot of glucose or it cannot be taken up or whatever, then it might be a good thing indeed, not being burned properly. So that might be how it works, but yeah, just to add on.
Great. Nice addition. Thank you, Miranda. Thank you, Job. I think we go ahead with the next speaker. So if there's a go
questions will still be answered in the chat, right, Roger?
Yeah, for sure. Then we the panel is still active, so we can always answer more questions if people have questions on the metabolism. So the sugar and the fat uptake, what Miranda was talking about. So please go ahead and phrase your questions in the Q&A. So I think for the last speaker of today, for the lecture of the day, so we have Professor Duvedans, and which is a bit difficult to announce because I think he's one of the frontmen of cardiology, also worldwide. If you're scrolling through his awards and publications, it's a never-ending list. So I think he's approaching the 1,000 paper publications. So that's an insane amount and a huge experience in in cardiology. Currently, he's a cardiologist at UMC in Utrecht, the Netherlands. And yeah, of course, when he can tell you about future perspectives of lemmonopathy, then he's probably the best to answer. So I'm very happy that Peter Duvedans is here with us today. So Peter, if you can start your slideshow, it would be great.
I'm trying to do so, but not successful yet.
Got the pause music for it will be for next patient meeting.
Yeah, that's a good one. Maybe I could do an extra question, a bonus question. I was wondering if I should ask Miranda if that's still possible while Peter is looking for the
Can you call me?
for the presentation. Otherwise, you can send it to me as well, Peter. If you send me the presentation, I can share it for you. That's possible as well. But just in the meantime, one more question. You mentioned having healthy balance, and it's different from person to person. Do you think that's going to be different in the near future, that it's possible to make it more personalized?
Ideally, I would say yes. That's but it indeed, yeah, it takes a lot of time. We need a lot of information there. We would need, at least at this stage, we cannot do it like just by imaging. It's so, yeah, ideally at this stage we have blood or biopsies to do it. And of course, it's quite a lot of work if we do it per person. I think what we want to start with is looking perhaps per mutation, because I also think within lemmonopathy, you have different mutations, of course, with different phenotypes. But ideally, I think, yeah, we would eventually like to go towards personalized medicine, because I think, yeah, it's almost impossible to do without.
Yeah, it's a bit related to the question that was in the chat as well. If it's more like to do with diet as well, if what works best for like certain people. So that's what I was wondering as well.
Yeah, I think, yeah, like Magdalena also said, but I think at this stage, a bit of a mix, not too much of one type of diet is probably the best. Okay. If you don't know your metabolic phenotype. Yeah, if you're diabetic, exactly. Of course, then yeah, it might be already a bit different. But
yeah, exactly. Okay, see how we're going with the next presentation. Is it possible to share from your side?
Something to add as well?
Yeah, and I also wanted to add to everything what Miranda said. Of course, we are measuring the mitochondrial loss that was, for example, shown, or glycogen surge at the end stage of the disease. But laminase mutation is given through the conception at the beginning, right? So actually, it gradually comes to that stage. And for example, now I'm thinking when there's less mitochondria, which is also seen in other cardiomyopathies, there's like a loss and damage. And basically now you have less carburetors to process the fuel. Then you know that you have to do everything to have enough of the fuel. And this is not established. We can't give recommendation like European or American Heart Association, Society of Cardiology recommendation at that point. But if you think in this beautiful example how Yolanda showed the motor and the fuel and how the car works, just try to feel. I experienced something like that as well in my lifetime. I'm not diagnosed with cardiomyopathy. I don't have any mutation, but have experience with this. And this actually I used my knowledge from the science and try to really make sure that I have a healthy amount of fuel. In periods where I didn't perhaps think about eating, and I experienced arrhythmias for many months, and they disappeared once. I'll make sure I don't start with 2 coffees, but I have like a healthy, balanced, breakfast. And this is very subjective. Don't take it as an advice. But I think in general, balance is something that you should think for yourself what feels good for your body.
Okay, thank you. Okay, how is it going with the presentation? Is it possible to share Peter's presentation already? Let me see.
Working. I'm gonna send it to Ilko.
Yeah, if you send it by email to Ilko or to me, Peter, then I can, or then we can share it. That's no problem at all.
Maybe I can also fill the time by other studies that were done on mice, for example, where from our colleagues in the Netherlands or worldwide, when for example mice were given omega fatty acids or not. There is a group with and omega and without the omega fatty acids that we know from Mediterranean diet, from in Finland, for example, people have for breakfast a spoon of fish oil. So these are foods, for fatty acids. And then researchers, for example, in Amsterdam made some time ago already created heart failure by constricting, like closing the vessel that is coming out of the heart, and created the heart failure based on that. The mice that had the support of these fatty acids did not die, and or as much as the ones without. And also other colleagues from Groningen have shown that other metabolic substrates, even preventively, can prolong when the disease starts. So there is literature and knowledge from mice, but that is not necessarily the same as in humans. And I would like, Miranda, can you give some explanation because you are expert also on mouse metabolism and you've done those studies, how do you see the translation to humans? And is this like one-on-one, or can we still learn from something even if it isn't?
Yeah, that's a good point, of course, always. Also because mice, they are much more, the mice that we study, they're usually, it's one family. It's really, they're very much the same. And also, I will make it very quick because I see the presentation is ready. But yeah, and the metabolism is different than in people as well. So but what we see is that if you give a mouse a high fat diet for a long time, or if you give it a high carb diet for a long time, we see cardiac problems appearing. And we see in a lot of cases, we see similar things occurring in patients that eat a lot of high fat or a lot of high carbohydrates. But there's also definitely differences between the mice and the patient, and also because, of course, the diet of a person is much more diverse than what we give to the animals as well. So
yeah, indeed. And the studies that I mentioned, this wasn't high. Huh? This was sort of like a normal supplement. So it really depends on if it's on the balance of how much you get. But high is perhaps too bad as well.
Yeah, too much of something is usually not good enough, I think.
Right. I think Peter is all ready to go. So the floor is yours, Peter. And you're muted.
Sorry for the delay. Thank you, ladies, for saving my life again today, for filling the gap. I think most of the things already discussed on the pharmacology, I think, are very relevant and also show the limitations that we currently face with treating LMNA patients in a very general treatments, I think, for heart failure in general and not so much specific for this disease. So that's why we try to develop something that's more specific for the disease and maybe even very specific treatment. And in the end, maybe even a cure for the disease. But I think that's still far off. So if you ask me for the magic bullet, it's just not there yet. And I got the slides from Yigal Pinto, and I hoped he had the magic bullet, but he doesn't have it either this morning in time, I think. So we have to wait and be patient and expecting what comes from the future. I will go into that a little bit more in detail. We do know that one of the treatments that's very essential in many of the patients with arrhythmias is implantation of an ICD, that's an implantable cardiac defibrillator, which is very efficient in preventing sudden cardiac death. If you look at a small study with 20 patients, basically, then almost 8 patients had, within 2.5 years, had an appropriate shock, which is really a very high number of patients. So it's a very adequate therapy. But we all do know that ICDs are, although they are very protective in sudden cardiac death, also have their problems, because it's not nice to have a device like that, and it's especially not nice to get a treatment from an ICD. So it's a nice step forward, but it's definitely not the solution to the problem. That's for patients with arrhythmias. Jesus, nothing is working today. So never take over the presentation from somebody else. And then we can, in the patient group at LMNA, we can predict basically based on risk factors, which patients are very prone for arrhythmias, which patients are a little bit more protected against arrhythmias. So here you see kind of a risk score. If you have an impaired ejection fraction or you have non-sustained VTs, then your risk is much higher. If you have several of these risk factors, than if you have 0 of these risk factors. So that helps us to predict which patients should go for an ICD treatment and which patients probably do not need any additional devices. In general, we see the patients every year. They come for electrocardiogram. We do make an echo every 2 to 4 years. That's kind of varying depending on the stability of the disease. And we start looking at the cardiac function at the age of 12 to 13 years in general, unless there are complaints. But then, of course, it goes more often and more intensively to monitor these patients. So that these are the general rules for looking at these patients and monitoring them, basically. I already mentioned the ICD is a potential treatment for arrhythmias. You also give medication to kind of protect the heart. But again, that's not specific, that's pharmacology. We do not have a very specific treatment at this point in time, just general medications that we think are beneficial in lowering blood pressure and maintaining cardiac function. And I think many of the compounds already mentioned in the discussion previously, should be tested at a short notice in an appropriate way to see if we can add something to this pharmacological treatment of these patients. In the extreme cases for heart failure, we have to consider mechanical support. That mostly happens in the form of a left ventricular assist device that takes over the cardiac function, basically. And eventually, many patients will have to go into the prospect of a cardiac transplantation, something we would like to prevent if we can find a treatment that's much more curative and at an early stage. We don't wait so long before you have the stage of the transplantation that's required. Now, there's something happening in this world which is really changing fundamentally the way we look at disease and also the treatment of disease, as we do see an enormous boost in gene therapy approaches. And then cancer is way ahead of us. If you look at this, basically the circle, you can see that about 67% of all the treatments are being developed now for cancer, and much less for other diseases, but also monogenic disease like LMNA. There's already 11% of all the treatments are being developed currently are focusing and trying to find a cure for these monogenetic diseases. So I think there's a lot of activity there. And the good news is that although it's still in progress, also there's a lot of money available from companies that do think this is the way to go to the future. So all big pharma companies also moving into the field of genetic and gene therapy approaches, basically. So I had a slide of this video which basically shows what Marlene already expressed. I will skip that for the sake of time. There are different levels where we can intervene. So we can consider, as Marlene beautifully told you, that the DNA is in the nucleus. Part of the DNA will be transcribed, that leaves the nucleus, goes to the cytoplasm, and there in the cytoplasm the protein is being formed. And we can interfere with this process of the transition from the transition to the translation by interfering with RNA molecules. That's called either antisense oligonucleotides, which are small non-coding RNA molecules, or with small interfering RNAs. It's just a different principle. It's a little bit more stable than the antisense approach. Also, they're both aiming to try to intervene with the translational process. So they block the messenger RNA, basically. Of course, other things that we can consider, there was already discussion on colchicine, which is basically blocking immune response. You can also think about blocking immune response in a more general way in many of the diseases, including all the cardiomyopathies, basically. And a very novel approach is using aptamers. It's kind of based on antibody, parts of an antibody to deliver specific molecules to the cell, to the cytosol. So there are different levels where we can interact. We can also try to go into the nucleus and there correct the genetic defect completely. There are different ways to try to reach a cell. This is just an example of using adenovirus. Adenovirus is very efficient in targeting basically all the cells, endothelial cells, fibroblasts, also all the cardiomyocytes, because all these cells have receptors that will basically take up the adenovirus and then bring the DNA, the content of the virus, into the cell initially, but also eventually into the nucleus of the cell, where it can be active for a short time if you use adenovirus. There are different viruses that we can play with and use for this kind of purposes, and they have different characteristics. Most of the studies now are basically really looking into the use of adeno-associated virus. That's the second column that you can see here, basically. But also lentivirus is something that can be considered as a gene therapy, not so appropriate for the heart, we think, but it's still an option. Adenovirus is a little bit bigger content, so you can put a lot of genetic material into the virus. You can package into the virus there. AAV has less capacity to where you can introduce DNA. Lentivirus is a little bit bigger. The good thing about adenovirus can be that it's episomal. That means it will be degraded in time. So it's not permanent. It will be just there temporarily. AAV is a different thing. It's mostly episomal, but also the part of it is integrated or connected to the chromosome, basically. So it will not be degraded, and it gives an expression for a long, prolonged time. It can be months or even longer than that, maybe even years. A lentivirus will be different because it's kind of built into the DNA, and that basically changes your genetic information. And also what you would go, if you have offspring, the same virus will go into the next generation then, basically. So that's a more tricky approach to try to do something into the DNA structure. So 3 different approaches with different advantages and different disadvantages as well. But they're all being looked at as kind of a way forward for therapy in cardiomyopathies. Now, this is one example of such a treatment for AAV. That's the adeno-associated virus being used here. But you have to realize for this unique disease, they basically charge $1.2 million per treatment. So this is a very rare spinal atrophy, dystrophy. So you have paralyzing of your muscles, especially in the legs and in the arms. It's a very severe disease. It affects young people, and they are all homozygous for the mutation. So that basically means that you get a wrong copy from both parents. In this case, it's recessive. So there is a therapy, but it's extremely expensive. 1.2 million is, of course, outrageous amount of money for one treatment that you need in this case. And we don't know exactly how long it will be beneficial and how strong it will be, because it's still very experimental. So, but this is a way to show that the companies also, that's probably one of the reasons the companies are interested in trying to embark in a project like this. Another approval of the gene therapy is in a different disease. That's in the amyloidosis. That's also a disease that affects the heart. It's kind of storing proteins in the heart, and it also causes dysfunction of the heart. And here the FDA, the Americans, approved first of the kind targeted RNA-based therapy to treat this rare disease. That's something that's been done like, I think 2 or 3 years ago, 2018. So this is now on the market as a therapy for amyloidosis patients, showing again that these kind of approaches are just really entering the clinical arena now and becoming a reality. Also, this is a very expensive treatment, by the way. Now, if you think about LMNA more specifically, then we have to consider either to try to block the progress of the disease. That's something we can do by interfering with the RNA molecules. There's not a cure, but it could be a method to reverse symptoms or block progression of the disease. And I think that's something that becomes a clinical reality within 5 years, because I think we are very close into looking at different approaches, and we have already some results in different diseases that show that this is probably going to be likely to be implemented before the 5-year term. If we think about gene editing, and that's basically correcting the mistake, that's basically we're using either TALEN technology or CRISPR-Cas or base editing or whatever, or prime editing. That's going to take a lot more time because it's far more complex. It's far more complex to deliver the therapy to the heart specifically. It's also far more complex because we don't know the off-target effects of the kind of approach in gene editing. So I think it's coming. Also, that's on its way, but it will take a lot more time before it goes into the clinical setting. It's feasible, has been shown by this publication by one of our colleagues who's now working in Utrecht, Francesca Silitano. She showed it's possible in another disease, another cardiomyopathy, to correct the mutation basically. Or in this experiment, they basically knocked out specifically the deleted or the mutated allele, basically showing that you only have the wild type normal gene that's expressed and you have a normal protein, no disease protein anymore, which was very effective in this study, showing that you can really use the technology, TALEN technology, to correct the defect. Also, if you look at functional data, they basically show that after correction, they have basically a normal force of cardiomyocytes. So also the phenotype after correction is completely recovered basically by this kind of approach. So I think that's showing just showing that it's feasible, in this case in isolated cells and engineered heart tissues, not in vivo. But also in vivo in the meantime it has been done. Looking at LMNA basically, that's already passed 2 times, that we have also different people that have progeria. That's a very severe disease where you age extremely rapidly. So kids can die basically of old age at the age of 6. We don't know exactly what's going on there, but there also is a mouse model for this disease. In that mouse model, they used AAV injections after 3 days after birth or 14 days after birth. And that's very effective in prolonging survival in these mice. So basically what they showed in this study is that if you do an early injection, you have a 1.8-fold increase in life expectancy in the mice. If you do a little bit later, after 14 days, it's even better. It's 2.2 times the survival expectation that you have there. And you have to realize that a mouse in a lab normally survives for about 2 years. So it's almost back to a normal life expectancy if you treat these mice with 1 injection of an AAV with the corrective DNA included in this construct. So I think this is very spectacular, also showing that in vivo can really change the cause of the disease indicated here. Now, as I mentioned, you can also play with trying to block the progression or try to reverse symptoms, indicated here by using antisense therapy and by small interfering RNAs. Just 2 different approaches. You can basically block the allele that's diseased, so that will not be expressed, you will not get a toxic protein. And in that sense, you can have enough of the wild type protein, normal protein, which can basically do its function, and you probably can correct the disease in that way as well for a temporary situation. It's not a permanent situation here. And one of the results that Yigal sent to me is this slide basically where they showed he used another approach, a short hairpin RNA, compare that to a scrambled RNA, and he basically shows in this slide that he can modify the nuclear form. He thinks that if you look at the right side, that the nuclear form is much better, showing that the lemon has a normal function again, recreating the normal morphology of the nucleus. So he thinks this is really a way to go forward, but it's just morphology. It's not really showing any functional changes yet. So I think that will be the next step to make sure that you also see a functional improvement here. So in summary, I think it's very important, I think it was also already indicated by Magdalena and by Miranda, that you really have to know the basic mechanism. Is this metabolism? What's the early changes? Is it the mitochondria? Is it something completely different? Is it the nuclear envelope that's really diseased? So that's something we really have to look into and try to understand. And I think that the gene therapy is becoming a real option for treatment, especially for delaying the progression of the disease. In the future, it may also be a realistic approach to find a cure for the disease, but that will take longer than at least 5 years, maybe even longer than that, because we really have to know the off-target effects if we are also interfering in an unknown region. I think you have to keep an open eye for apparently unrelated therapies like the SGLT2 inhibitors already discussed in this discussion. But also colchicine was in the Q&A question. Apparently there's a study ongoing with colchicine, which is blocking inflammation, a very general approach. This can also be a very nice therapy for the future. We also recently showed that you can block coronary artery disease using low-dose colchicine as well. So it's definitely worthwhile to also pursue this in this disease, where blocking inflammation in general is a beneficial approach. And that's also bringing the old-fashioned drugs that can be developed and will be developed, maybe more affordable than the very expensive gene therapies that we are discussing at this point in time. So I think there's hope in the future, but at this point in time, there's not really a specific disease treatment yet. So we have to keep on working together with the patients, because I think that's very important to work with patients to make sure we do the right thing and we develop the right therapy for the right people and we give it at the right moment to be successful in trying to find a solution for this disease. Thank you very much.
Great, thank you, Peter. It's a very nice overview, and it's good to know what the scientists are working on to have some clear perspective for the future. There are some questions already. To what degree does family history of tachyarrhythmia predict the likelihood of arrhythmia as an early symptom of disease?
It's not in the risk score that they use at this point in time, but we assume, of course, that most of the people with a LMNA mutation do have a family history. So in that sense, it will not make a big difference in the risk score approach.
Okay, I have a general question as well from own part, because I get this question a lot of patients. So you were mentioning adenoviruses and adeno-associated viruses. So would the virus harm in any other way? Because a lot of patients are a bit scared of using a virus as a therapy because of the whole COVID situation right now. So can it do other damage?
Yes, it can. So the problem there will be that we have to find a solution to that, because if you give a very high level of either adenovirus or even adeno-associated virus, there's a maximum that we can give without damaging the cells, basically. So we really have to be careful with that. That's why we think we now have tools to deliver the gene to the right cell, but the delivery as a whole, only to treat the heart and not the other organs. If you really want to treat the heart, that's still a very difficult thing to do. So delivery specifically to the one organ, that's something that's a big challenge at this point in time. That's something we have to solve before we can go to clinical trials anyway. So too much virus is detrimental for the survival of the cell.
Another question, it feels like, as in many disciplines, that we're all feeling a different part of the elephant, as it were. What are the institutional hurdles to a multi-drug trial in the Netherlands?
I think to a multi-drug trial?
Yeah.
That's a difficult question. I don't understand the question exactly. How do you want to do a multi-drug trial?
So that I think the question is, can we, what kind of trials are currently going on in the Netherlands, and can we use a combination of those?
Okay. So I think for the patient population that you have now, one of the key things will be to work, I think, with as much possible human models. So with IPS lines, with engineered heart tissues, with tissue slices, and then test different compounds, because there are many compounds that may be beneficial. If you look at metabolism, we can think about 10 to 20 different hypotheses we can test to see how functional and how important they are. And that platform you really have to use to make a selection of what you will bring to the clinic. And I think then if we go to the clinic, then I would like to have an international trial, not a Netherlands trial. But I think you directly have to make sure that you do an international study. And I think it's more important to find patients in the same stage of the disease internationally than try to do a local study in one country, actually. So I would opt for European or even global studies, actually. But this platform...
I completely agree. Yeah. Yeah, and that's also one of the benefits that the LMNA Cardiac can help in, to try to assemble all the people, all the patients with the same stage of disease. So that's it's a very nice point. Let me see another question. Does the AAV vector deliver the entire correct LMNA protein, or does it deliver the correct letter, so the amino acid, to allow the heart cells to make the correct LMNA protein itself?
Yeah, so the option of delivering the LMNA gene directly and then make the protein That's an option that's temporarily beneficial. If you think about curing the disease, you really have to cut in the DNA and make sure you take out the mutation and put in the right base, basically, which is a different approach. So you need different tools to make that happen. You need CRISPR-Cas technology to introduce it into the nucleus of the cell. And then if you want to do that in lemon mutations, you have to introduce 3 viruses at the same time. So you cannot do that in 1 virus, it's too small. If you want to bring into all the tools, you have to deliver 3 viruses per nucleus in each cardiomyocyte, basically. But it's far more complex.
Another question, as patients, we often need to advocate for ourselves with our clinicians. How can we best stay informed and provide relevant information to our care teams?
Yeah, I hope that this foundation, your foundation, Stefan and Rogier, can do that basically. That's, I think, one of the tasks and one of the beautiful things and one of the challenges that you are facing, to make sure that you provide the right information to your clients and that make sure that they can help the physicians in making the right decisions, basically.
That's basically how we started as well, Peter, because I started doing, I was just Googling a little bit on LMNA and I couldn't find anything whatsoever. And that's where we're providing a little bit of information on LMNAcardiac.org. But our main goal is to build a strong network and speed up research and development. So that's definitely our main goal. But with these kind of webinars, right now we have an LMNA patient meeting. Last time we had an LMNA expert meeting. We're trying to bring things further as well, of course. But that's definitely one of our main goals. But our main goal is definitely building a strong network, speeding up research and development, and hopefully finding a solution for it. But it's definitely true. There needs to be more information available as well. So if there's I'm not quite sure, Rogier, what do you think on the information side?
Yeah, I think so as well. So I think these meetings help, but also to connect the right patients to the right people. And if they have certain questions, we are able to direct them to the proper place instead of having, for example, metabolic questions to people not familiar with the field. So I think we can direct them. So if there are any questions, anytime, people can mail us at the LMNAcardiac.org. And we will help you answer any questions or direct you to the person who knows best.
Yeah, if you want to get connected with someone, or if there's a new initiative, or if there's any questions like the one with Miranda just before, with the question that you guys have on metabolism. So it's definitely something that we are more than happy to help out with, and hopefully speed up things and bring things a little bit further. So
yeah, I think it's nice if you can find a physician that has a hobby in LMNA mutations and has at least some interest in trying to deep into the heart of the matter, basically.
We have more questions, actually, Peter.
Quite a few.
Why does the LMNA gene increase morbidity in heart failure patients?
Yeah, that's the key question. So basically, we think that it has an effect on the nuclear envelope. That's highly likely and also shown, I think. But that does not mean that we understand what's really happening there. So if the protein is not functioning properly, how does it affect nuclear function? Is it just nuclear function? Is it something with DNA? Is it something with metabolism, with the mitochondrial activity? I don't know the answer. Maybe it's a combination. If you have 2 hits, that you have more disease than if you have 1 hit.
Mechanical stress as well. Yeah. Let me see other questions. I noticed ICD implantation. My dad died 36, sister 42, no ICD implants. Yeah. I have a cardiac scar and ejection fraction of 60-65. I have a missense mutation, not male, and was implanted. I appreciate this VTs. So will the criteria change? So one get implants before death.
Now, I think if you have VTs, that the threshold to implant an ICD is extremely low. So I will be surprised if you find an interventional cardiologist or an electrophysiologist who's not going to implant an ICD if he does see VTs. I think that's a very strong indication, because then you're in the high risk group.
How can we get involved in research as patients? My whole family with many affected individuals will be supportive of whatever we can do to promote research and treatments.
Yeah, I think that's great. I think that's just what we need, because I mentioned already this platform. I think in this platform you need different phenotypes, different genotypes, because you have different mutations in LMNA. So you have to collect all of that and make sure that you have, maybe not in one place, but that you know where you can find the different mutations, the different lines of IPS lines in the heart tissues, that you know where you can find them. And the patients, I have to get into control of this whole process, basically. We just supporting the patients, not the other way around.
Yeah, and indeed, in terms of privacy, I won't mention the names itself, but it's families with lemmonopathy is extremely important from both genetic point of view, as clinical point of view, as metabolic point of view. So see where is the closest university medical center, and see and put your question there, because it's extremely useful for research to have large family cohorts with people that have the mutation, but also that do not have the mutation, because you share a lot of similar genes. And if you have the mutation, and perhaps you have the mutation, but you are symptomatic or asymptomatic, so that's a huge resource. So it's very important to offer this, offer just a tube of blood to that research facility. So I think it's great that you want to help as a patient, and this is really great. So please do so.
Yeah, and please reach out to us as well, and we can see how we can help in that particular situation.
Yeah, and if you need someone in the network that's going to help you, then we can look also for the person who's going to help you in the network to make sure that you do the right thing in the right biobanking approach. I think that's crucial also.
Yeah. Send an email to LMNA Cardiac, and we'll guide the way. Let me see. What percentage of known LMNA patients require a transplant?
That I do not know. I do know that in Utrecht we transplant them on a regular basis, but I don't know the exact pool of LMNA patients. I don't know how many people go into the heart failure and the transplant program. I have no clue. I would estimate something like maybe 5 to 10%, but that's just my thumb.
Yeah,
How many?
I have no data.
I know the latest publication is from 2018. It was a really large cohort of LMNA patients from Sweden. And there the prevalence of heart transplantation was 19%, so 1 in 5.
Okay.
But it's, yeah, it can be completely different in different cohorts. It depends on the mutation, it depends on so many factors. So it's not just a clear answer, but there's about 1 in 5 to 1 in 10.
Another question, why is it that some members of the family are mildly affected and other is severely affected?
Yeah.
How is that possible with the same mutation?
Yeah, I go back to this two-hit approach that I think if you have mutation, you're more liable to get problems. If you then have something else, maybe a viral infection, maybe exercise, maybe something in your metabolism, something else. If you have a second hit, then you will get into trouble. And if you do not get the second hit, then probably you're protected, or it will be delayed the onset of the disease, basically. But maybe Job has a different perspective there.
Yeah, it's definitely, we think there's certain triggers that some people are exposed to and other are not. We don't know exactly what these triggers are. It's also definitely some the genetic background, because you get a genetic background from your father and from your mother, and you get a mutation from one of the two, but from the other parent you get another genetic background. So it's not only that specific gene, but all the other genes you get. And some of these, this genetic background can be protective. So you do see differences in families, because there is every time there is a dilution of the genetic material. But also this part, we are really at the beginning of investigating this, but we are quite convinced that it has a great influence on the differences in families.
In another disease, we are looking into the effect of the environment. If there is pollution, does it have an effect on your cardiac function? That's something we are looking into for another disease. But I don't have the data yet. So it's just something, all the options are open, basically. Everything's on the table. We don't know.
And I have another question. Today, a few times LMNA mutation and exercise came up. Is it okay to play very intensive sports with an LMNA mutation showing minor symptoms like AV2 block and NSVT, but no scarring?
Yeah, I find it very hard to give a strong statement here because I'm a sports fanatic myself. So to discourage people to go sporting is something that I find very hard to do. But I think you have to be careful because your heart is more susceptible to damage than the heart of a person without a mutation. So maybe you do have to take some constraints actually, but I hate to say that. And I don't have strong proof either. This is just a hypothesis. It's just like being conservative as a physician. I think that's more the background there.
Yeah, I recognize that. It's a very difficult statement as both sport fanatics and but it's very tricky. I'm used to play volleyball in my case as well at a high level, and it's difficult to say, but just be cautious there. Just another question came in. An older generation of family, 4 of 6 siblings are LMNA positive. Next generation of 16 first cousins, 50% are positive. Third generation continues carrying the disease. Of the 4 older siblings, 3 have passed. Most were treated with amiodarone to regulate cardiac function. Mobility has severely deteriorated, probably due to amiodarone. Do you have any preferred therapy that maintains mobility as much as possible while controlling heart function?
Yeah, so that's a very sad story of about, I've been in the field for 25 years. We have been looking for antiarrhythmic treatment by using drugs and have been extremely unsuccessful. So basically none of the drugs has been on the market is still there, except a very old one, that's amiodarone. That's been there forever and it's still there. That's the only one that's really working. So that's a bad and sad conclusion, but we don't have anything else. So if you need amiodarone, then you should also consider to use maybe get an Icd and try to use a beta blockade or something like that to prevent the amiodarone side effect. Because they can be very severe amiodarone, especially in long-term usage. You get a neuropathy, you get problems with your thyroid function. So if you can get away from amiodarone, then that's a very good thing to do actually. But I'd probably have to get an Icd then.
There are 2 old questions, and I don't know if they are answered, but let's treat them anyway. What determines how severe the LMNA DCM disease becomes?
You mean if you can predict the outcome? That's very hard to predict anything.
So what is determining factor how severe the DCM becomes?
No, I think there will be differences in mutations. So in the whole gene, there are different spots that cause the cardiomyopathy and also different kind of mutations. So that was addressed by Magdalena very well, I think. That plays a role. So the mutation itself plays a role. And then again, how do you go through life also plays a role. So all the things you run into, viral infections, myocarditis, whatever, the same story again, that will basically determine the speed of the disease, the progression of the disease. There's not one thing that you can pick out and focus on.
Do you know if the medicine Entresto can slow down LMNA DCM?
I do not know. I know it's a very effective drug in treating heart failure in general, like many of the drugs we discussed this afternoon. But I have no specific data on Entresto. Job, do you know anything on that?
No, I think it's the same as the SGLT2 inhibitors. It's relatively novel. So we don't have any data yet on specifically for LMNA, but it will be really interesting to collect the data.
If you have a platform, you can test it in different cell lines. That would be the way to go, and then you can move forward with clinical approach.
Yeah.
Worth to actually do that. Yeah. What are the current treatments for LMNA DCM as it progresses? I have an Icd and I'm taking beta blocker to help with arrhythmias currently. Are there any treatments that are not recommended that may be used for other heart failure patients?
Yeah, I think in general, I think that an ACE inhibitor or an AT2 blocker will be something you should consider, unless you have a very low blood pressure. You cannot cannot take it because I get complaints from low blood pressure. But otherwise, I would always take an ACE inhibitor actually.
Let me see. 6 out of 6. Let me see if I understand this question myself. Let's see. So I think what she means is there are 6 of us siblings, age 52 to 61, have LMNA with limb girdle muscular dystrophy. At 61 years old, I feel I'm losing muscle and do have some pain in my hips and weakness in arms. I do exercise mildly, and I have done so since very young. Does working out waste my muscles, but I can't rebuild as normal individuals?
I do not understand the question either completely. I'm reading it, but I do think whatever you do, if you take mild exercise, it's always good. And if you stop doing it, you will waste your muscles even faster. So I would definitely try to keep it up, actually, to keep your muscles in shape. So
exactly, that's how I read the question as well. I think it's you should remain exercise. So she says she's already exercising mildly, so I think that's perfect. And I think it's perfect to keep on doing that. Indeed, as Professor Duvedans said, if you just stop doing that, then you would waste muscle anyway. So I think it's very useful to keep on exercising mildly, walking, cycling. Are doctors taking ICD implants seriously in LMNA patients or just males? Is that changing? Oh, let me see how to read that.
Yeah, the risk for male is higher than for female, maybe. Yes?
Yeah. Conflicting information as patients seems. We have to advocate for this, and often doctors are taking LMNA seriously. I think in increasing matters, then doctors are actually recognizing LMNA. LMNA is a fairly young disease, so it's so we even saw different disease phenotypes. So how the disease actually occurs. We see in the last few years there are several diseases that we now can connect to LMNA, and so it's a very young disease. In that sense, that we start to understand a little bit of LMNA. So I think doctors are taking it very seriously in increasing matter. So I think that's a positive thing.
But I think in the Netherlands, I don't know about the other countries, in the Netherlands you're entitled to a second opinion. So if you're not happy with the way the doctor is treating you, then you're entitled to ask for a second opinion from another doctor. And I think also there, as a foundation, you can basically help make sure that you have the doctors in the area or in the country that people can refer to for a second opinion and ask another opinion from an expert there, if necessary.
Yeah, I agree. And I think it's an old statement. So within my own family, they also used to say, like 20 years ago, they said, okay, it's just male or just female, but now we know it's not the case. Yeah. So that's happily we improved in that. What is the impact of alcohol and caffeine on progression of disease?
Yeah, I would think if you are moderate in use, that it has no effect. And then moderate in use, I would say that you take maximum 2 consumptions of alcohol per day, and like maximum of 3 to 4 coffees per day, or also tea has some effect there. But if you take it moderately, I don't think that will have an effect. Also, that's not proven. That's my gut feeling.
Another question is, I take a beta blocker and not an ACE inhibitor, but an angiotensin losartan. Is that okay? The Mayo Clinic said as a trial to slow progression of heart failure.
Yeah, I think that ACE inhibitor or 82 blocker, I mentioned that already, are very useful in this case. So I don't think that's really important if you get one or the other. They're both effective.
Another question is, my wife has LMNA cardiomyopathy and was prescribed Entresto by her team at Stanford University. They shared their LMNA patients were responding well to it, and it has become a part of their treatment protocol. So I think that was what was answered before, I think by Job, the Entresto treatment.
It would be nice to get the data. So it would be nice to talk to the doctor who is doing that and looking at the data. Because that would help you as a foundation as well to make sure we give the right advice to people, because I'm not aware of the data.
We should write this one down, Rogier, and get in contact. We have contacts there anyway, so we can definitely get some information.
Yeah. Luckily, with some people, we are here at the expert panel. There are a lot of contacts with Stanford University, so that's fairly easy to check up.
But
so
in general,
we will.
In general, I anticipate that many of these drugs that are now being used for heart failure in general will also be beneficial in cardiomyopathy patients. There's not a specific treatment, but that doesn't mean it's not a good treatment. So if it's helpful and you get reduction of symptoms, then it's fine, of course. But it's an SGLT2 inhibitor and Presto, it doesn't matter, of course. If it treats heart failure, then you're going to be improving.
Would you recommend IVF and genetic therapy to make sure LMNA is not born by embryo and thus stop progression to next generation? Yeah, so would you recommend IVF or genetic therapy?
I think I will refer to Miranda, I think, because probably she's in Maastricht and knows everything about this kind of approach.
Yeah, maybe I can answer that question because I work on this topic also specifically. It's a very personal question. It is possible. Of course, it's the only way to make sure that you won't give the mutation to your next generation. But keep in mind, it's a very intense procedure. It's embryo selection in the laboratory plus an IVF trajectory, which takes in total between 1 and 2 years without guarantee that leads to a pregnancy. And it's also, it's a quite intense trajectory. So it's a personal thing if you want to weigh the efforts to, how much you would prefer to not give the mutation to the next generation.
And Job, is that an accepted therapy in the Netherlands at this point in time for LMNA?
Yes.
Okay.
Yeah, it's pre-implantation genetic testing, PGT we call it.
I don't know if I can say that, but I have a very healthy niece because of this procedure in Maastricht. So that's, yeah, it works. Is there any known association between NKX 2.5 and LMNA mutations? Some members of my family have both, unfortunately. Have you seen any? Have you seen such patients in the Netherlands? So NKX 2.5.
Oh, NKX 2.5. Not with LMNA, no. No, not that I'm aware of, no.
No, in my biobank, I don't have NKX 2.5 and LMNA at the same time, no. Yep.
No, it's always a combination of two mutations. It's also always very rare. And we have some combinations of LMNA and Titan, for example, families, but this specific combination, we don't have any families. So
I've seen LMNA with myosin heavy chain. I've seen LMNA with Titan, indeed, LMNA, and yeah, but I've seen several with combinational mutations, but not with NKX 2.5. It's interesting because here we're talking about developmental biology, so that's actually interesting. Yeah. Has anybody on the panel ever any comments on studies showing oral nicotinamide ribosome consumption to boost NAD+ levels in cardiomyocytes to boost cellular energy, showing improved heart function? I think we go to Miranda then.
Yeah, I was afraid of that, but yeah, I don't know at this stage, to be honest. I know there are studies going on. I think there are some things published there. And I think some of them are beneficial, but I don't know enough about it to really draw a firm conclusion at this moment on it.
I have on NAD+ levels, I have a very nice collaboration with Antoine Mouchier in Paris.
Oh, wait.
And so with a collaboration with Paris, and they are 100% convinced that it should increase heart function. Is it published? Well, there is a publication on NAD+ and NADH on heart function, but is it specifically an LMNA? I don't think so. But if you send an email to LMNA Cardiac, I will send you the publication from Paris showing on NAD levels. So there is some research going on that, and we are currently testing it on LMNA lines as well.
info@lmnacardiac.org is the easiest email address to address it to.
Hi, probably is not a properly question. I'm affected of Emery Dreyfus type 2 with an LMNA mutation. Could I ask, what are the expectancy of life?
I don't know.
I think that's very hard to say because we see a lot of variety in life expectancy also with Emery Dreyfus type 2. So it's...
Isn't that a good question for one of the other groups as well that are more into information on that? What do you think, Rogier? We have context there as well, isn't it?
Yeah, I can refer you to a group in Italy that is specifically on Emery
exactly.
Dreyfus.
That's the one that I meant as well. Maybe that's good to get in contact there.
Although with this limited information solely that you have an LMNA mutation leading to Emery Dreyfus type 2 is they will not give you an answer on that because there's so many factors play a role in this. So it's very hard to just point out the number if you don't have any medical information.
Yeah, send an email as well and get in contact with the group in Italy, I'd say.
Not for the entire call. Contact Dr. Ewan Ashley at Stanford University. Yeah, on use of Entresto. So that's then for Peter and Job. So Ewan Ashley, familiar to you?
We get the list of questions after the Zoom meeting as well, right? So this is things that we can take.
So apparently this is the physician at Stanford University that is.
I will write it down right away to get in contact. So that's great information. Maybe that's a good link to because I think this was the last question. We have a lot of questions. I think we have 44 questions in these 3 hours right now. If there's any other questions, just reach out and we can maybe handle some more questions, and otherwise just reach out to info@lmnacardiac.org. But the point I was trying to make, if there's any more information or if there is any other things that we can help with the foundation, just reach out because we're just trying to build the bridge and help out wherever we can. So happy to do so.
Yeah, perfect. And thank you for all the questions as well. So you've seen Job van Donschot already a few times answering difficult clinical genetic questions, and Job was actually awarded a very prestigious prize last year, or actually several prizes. And we will give him the opportunity to show you just a brief summary of his work. So Job, if you are able to share your screen already.
Yes, let me share my screen. Yes, I think my screen is now visible. I just have a very short presentation to show some of the highlights, and I think it's also very relevant to a lot of questions that came by. So how important it is to have patient involvement in research, but also the importance of patients willing to participate in research. So what you can see on my first slide is actually also the cover of my thesis, so of my PhD dissertation, which was about, the difficult title was Causes and Consequences of Dilated Cardiomyopathy. But let's see if I can go to the next slide. It's basically the main topic was about clinical consequences of genetic mutations in patients with dilated cardiomyopathy. So patients who have the disease, then we perform genetic testing to see if they had a mutation, yes or no. And afterwards, I try to characterize the disease as good as possible to get new knowledge about specific gene mutation and how relevant it is to the disease. So I defended my thesis, you can see here, last year, and that was in the middle of the COVID pandemic. So this is a picture of my PhD defense. It was quite lonely behind my desk. And afterwards, the defense was via Zoom. And afterwards, I could pick up my my PhD diploma, and it was also completely in the atmosphere of the COVID pandemic. To give a short overview of my research, it all starts with the most important knowledge that we have, and we call it the Maastricht Cardiomyopathy Registry. It's a large patient registry where we collect clinical information of patients. So we see them at the outpatient clinic in a hospital, and all patients get all the investigations that every patient would get. So an echo, an MRI, all patients are referred for genetic testing, screening for arrhythmias, and then they stay under follow-up at the DCM outpatient clinic we have in Maastricht. But we ask all the patients if they are willing to participate in research, and that we can collect all this information we get from these investigations in a database. So at the moment, we have about 1,500 patients with DCM in this patient database, over 10,000 echoes, complete genetic testing. So this is the only way how we can learn a lot about genetic diseases on the heart, by collecting a lot of information from a lot of patients. And Ilko also already said it earlier, another really important thing is the heart tissue bank. So the heart tissue bank is also a database. It's a biobank which collects the tissue of patients, and also linking this clinical information to the tissue information. That's crucial information to get to know the consequences of a disease. So the main findings of my thesis was that for 3 specific mutations, specifically Titan, filament C, and LMNA, I could better estimate on what age disease would start, how the disease course would go, and how it can be relevant in families. As Peter, Professor Duvedans also showed, is that in clinics we often use with risk calculators. So for example, for Icd implantations. In my thesis, I also tried to create risk estimation for patients with specific genetic dilated cardiomyopathies. And the last topic is a question that I get in the clinic a lot, is always from family members: what is my risk for developing dilated cardiomyopathy when I have the mutation? So also on this topic, I spend a lot of research. And this is still one of my main topics of research at the moment. So broadly summarized is what I did. I brought together information from the genetics and the cardiology. So the clinical, the genetic information we have from the patients and from the families, plus the clinical information we have, and with the eventual goal to get to know the underlying disease process of specific gene mutations to develop treatments. Then, as Rogier also said, after my thesis, I was awarded by the Association of Clinical Genetics in the Netherlands the Dissertation Award, the Benter Haar Award, which is a very nice award for young researchers who performed important clinical research in the last 2 years. But as I showed on my previous slide, it was not only genetics, also cardiology. So also from the Dutch Association of Cardiology, I received also the award for best thesis of the year. So it was really nice that I got the appreciation as well from the genetic side, as well from the cardiology side. Then, as I said earlier, my main topic of focus is now focusing on why are there differences within families when you have the same specific mutation in families, but there are so many differences between family members. So this year I was awarded a big grant by the Dutch Heart Foundation, specifically for a project focusing on this topic. This project was also supported by LMNA Cardiac. So this also shows that it's really important to have patient involvement and to collect data from patients to be able to answer these questions. On this slide, I also put my email address for if there are any questions or you would like to get in contact for this project or other research projects, then don't hesitate to get in touch.
Thank you, Job. Well done. I think it's great, and it does need my approval. You already got all the awards. It's really great and exciting work that you do. Patients, please reach out to LMNA Cardiac, and you can reach out to the panelists as well. Job just gave you his email address as well. In the Q&A, we have a big thank you, and I want to give that thank you to all our panelists of today. So I want to thank Magdalena, Miranda, Job, Peter for helping us out today with your expert vision, opinion, and the presentations that you gave. I think it's great to actually inform the patients, because the patients actually have the disease and they are worried, and it's very valuable that you give the time, your time to them to actually answer all these questions that were here today. So it's a great thank you to all of you. And I think we're about to finish up this patient meeting. So I want to thank all of the patients that registered and subscribed today, that were attending today, but also the people that registered and asked for the recording. So the people will get a link in their email for this recording of today, so they can see this session in their own time zone at appropriate times. So I can imagine that at 3 a.m. not everybody is willing to get out of bed, because we learned from Magdalena that a healthy sleep is also important. So that's very nice. I want to thank you all. I want to thank Netherlands Heart Institute as well. And Stefan wants to add something?
No, I just wanted to add that we will send out the recorded version as soon as we can. And there's a lot of questions already if people can look it back, but that's definitely possible. So we'll send it out as soon as we can.
We will.
And maybe one extra addition there, if someone wants to watch back the LMNA expert meeting, that one is online as well, because that's the meeting that we had in April. That's possible to look back the recorded version on lmnacardiac.org as well. If you can't find it, just reach out.
Thanks again, everybody. Thanks, panelists. Thank you, Netherlands Heart Institute. And we hope to see you all next time again. Thank you.
Thank you. Bye-bye.